Hi, everybody. We're here today for the next episode in these reflections on the history of modeling and theory. And these are hosted by the Society for Modeling and Theory in Population Biology and aim to really have conversations with the people that have shaped our field and witnessed changes over the last 50 years. I'm going to also thank James O'Dwyer is here with me, and
Hi, Sally.
hi, Kaylee Charlebois, who's helping with the technical behind the scenes work. So today we're welcoming Dr. Marc Feldman, Marcus Feldman. He received his PhD in 1969 with Sam Carlin at Stanford University. He's Australian, as you'll tell from his accent. And he had a brief appointment with La Trobe University in Australia, but came back and started as a professor in 1971 and is still an active professor, the Burnett and Mildred Finley Wohlford Professor in Biology at Stanford. He's had an enormous influence on our field. He was the founding editor of Theoretical Population Biology and served as its editor, I think, for 40 years, as well as serving on the board of many other institutions, including the Santa Fe Institute for the Study of Complex Systems. Marc is best known for his work on modifier theory, everything from the evolution of recombination, sex ratios, mutation rates, what have you, and on his work on cultural evolution, where he really laid the quantitative foundations for understanding the mathematical structure of transmission and selection acting on cultural traits. That led to one of his seminal books on cultural evolution with Luca Cavalli-Sforza. So we could measure his papers based on the number of papers. I think he's had over 600.
That's quite a lot.
I know, nearly 100,000 citations. But I really want to emphasize his impact as a mentor and supervisor. And I have to say this is somewhat personal because Marc was my supervisor as an undergraduate. As a freshman, I was introduced to Marc, and it really was through that interaction that I fell in love with mathematical modeling and evolution. I remember he got me hooked right away. He introduced me to a system of clam shrimp, Illuminatia texana, that have three sexes.
Ha.
And I was like, what? What's going on? What's keeping the males in the system? What is their frequency? All those kind of questions. So he got me hooked in as an undergraduate into modeling, and I stayed as a PhD student there. And I think Marc has had more students and postdoctoral fellows than any other professor in evolutionary biology, and he's had this enormous influence on the field. I got to share something. Let's see if I can do this. Hold on a second. All right. This is from Lee Altenberg, and it's one of these genealogy maps of Marc Feldman, and he was a student of Sam Carlin. Hey, look, it goes back to Fourier and Gauss. Really an incredible Bernoulli,
Wow.
an incredible pedigree. But this is out of date because this is only up to 2007. But 32 students. We'll have to ask Marc later exactly how many students he's had. But, I think it's, over 50. And then we're talking about his grand students and great grand students. And you're talking about an incredible influence on thinking and work in this area. And, one of the things Marc brings both rigor and a sense of artistry to modeling. And I think he elevates both of those aspects, the creativity, the insights, that aspect to models, as well as really kind of requiring high levels of rigor among his students. So he's had an incredible influence on me, and I without him, I can't imagine where I'd be, as well as the field as a whole.
Yeah, and I don't want to embarrass Sally too much. Obviously, your name was on that family tree that we just looked at, but the influence is really clear when you look at where those students have gone on to and the kind of work they have done and the influence they've had. Like you said, the academic grandkids and beyond. And so that influence is really huge. So it'd be really cool to see that tree spread out even further. Maybe we can find an updated one at some point.
Right.
So my definitely encountered his work very early on in my own career in mathematical biology. I really remember I was at the University of Oregon and talking with Brendan Bohannon. He had co-authored with Mark a really cool paper in ecology on rock, paper, scissors in a real system, in the real world. And so that was a really cool introduction. And then later, when I got to the Santa Fe Institute, Mark's influence loomed large. The ideas of cultural transmission, certainly, that Sally mentioned, and also niche construction were really kind of in the air at the time. And Mark's influence was really important there. Mark's awards were many awards. They include the Dan David Prize for outstanding contributions that shed light on human history, Lifetime Achievement Awards from the Society for the Study of Evolution, and also the Society of Molecular Biology and Evolution. Mark's a member of the National Academy of Sciences, the American Academy of Arts and Sciences, among many other prestigious scientific organizations.
Welcome, Mark, to the series.
Thank you.
Hi.
Thanks, James.
We're going to launch off with a question about your introduction to math as a tool in biology. When did you first realize that the two fields were as connected as they are?
After I finished my undergraduate major in math at the University of Western Australia, I started to work for IBM in Melbourne. That I had been in Perth at that time, which is a long way from Melbourne. And I didn't particularly like the company. The job, when I took it, looked like it was the best one in Australia. But so I started to look around for graduate programs. The math major that I had gave me some interest in pursuing a degree in statistics. And so I contacted University of Melbourne, and there was a new university that had recently opened in Melbourne, Monash University. This was 1963. And I wrote to them, and I got a very nice letter back from a wonderful guy called Jeffrey Watterson. And he said, why do you want to do this master's part-time? You can do it full-time, and I can offer you a salary exactly 1/3 of what you're getting now. I thought about it for about 10 minutes and said, okay. And Jeff had recently joined the faculty at Monash. He had been on a postdoc, having done his PhD with Moran in Canberra. And Moran, an illustrious figure in not just Australian statistics and mathematics and probability, but worldwide. And Moran had, as I found out later, strong influences on Warren Newlands and many other pure and applied probabilists in the Australian context. And Jeffrey was very interested in what came to be called later direct product branching processes. That was the term that Carlin introduced after he had seen what Moran had done and what Jeffrey had done on these things. And that Jeffrey gave me a problem. And it was clear that the math that I'd had before was very useful for these kinds of evolutionary problems. And Jeff was an inspirational guy, very relaxed, laid back, and a wonderful mentor. And the master's problem that he gave me actually eventually got published, and next year will be the 60th year of my publications. So
wow.
That came out in 1966, but it had been done in 65. And Jeffrey said, there's this guy at Stanford who seems to be interested in applying math to biological problems. Why don't you send your thesis to him, try to see if it'll get published in a new journal, pretty new at that time, Journal of Applied Probability. So I did, and Carlin sent me back a letter and said, why don't you come and be a PhD student? We've got this grant sharing with Professor Walter Bodmer in the genetics department in the medical school, and we can support you. So the rest of that is rather history. I didn't know Stanford. I didn't know Carlin, and it kind of forced me to read what they had done, what Carlin had done. And clearly he was a very great applied mathematician and had done a lot of work in statistics. And so that was how I accidentally parlayed what I had done as an undergraduate into the rest of my career. Thanks to Jeffrey. And then, of course, Carlin became extremely influential. And if you were to have polar opposites of style of life and research, it would be Watterson and Carlin. And so
maybe that's a good point for me to ask. So Jeffrey had this huge influence, really bringing you to biology early on.
Yeah.
And you've mentioned a couple of other names. Were there other big figures for you who really inspired you at that early career stage?
Yeah, well, when I got to Stanford, Walter Bodmer was extremely influential. You know that he had been a student of R.A. Fisher, had joined the genetics department in the medical school under the chairmanship of Lederberg, and was already a prominent figure in not just population genetics, but human genetics in general. And I was really, I would say, extremely fortunate that the two of them, Carlin and Bodmer, had put together this system that allowed me to interact closely. So in the, I think it was the third year of my PhD program, I took the first biology course that I'd ever had in the genetics department, and I was hooked. And after that, I took Charlie Yanofsky's course in genetics and molecular biology in the biology department and a few other courses. But Bodmer was very, very influential in became part of my committee, the thesis committee. And in fact, I wrote a paper with him before I wrote anything with Carlin. And that was on the RH polymorphism that he had mentioned in one of his lectures. That was a lot of fun to work on that. And it's been really fun to stay in touch with Walter over all these years after he left Stanford and went back to the UK, which had the sad but fortunate occurrence of Cavalli-Sforza coming to Stanford. So, I mean, it's absolutely clear the influence that Cavalli made on my career and life in general.
What a lucky circumstance for you to come, a magical letter that appeared welcoming you and bringing you into both a genetics and such a strong math training.
Yeah. Yeah. The aspects of chance in this trajectory never ceased to amaze me.
I hear you. Mark, you've made several discoveries over your lifetime, but is there anyone that stands out as a particularly exciting aha moment, something that changed how you think about things?
Well, after I finished my PhD, I went back to Australia for a number of personal reasons and joined the faculty at La Trobe University. Warren Ewens had actually convinced me to take that position there. While I was there, I pursued an avenue that had started with Carlin recommending that I read a paper by Masatoshi Nei, a 1967 paper. And I couldn't understand the mathematics behind it, behind what they had done. And that led me to try to reformulate what he had done in modifier theory in more precise mathematical terms. So the results that came out of that, I was surprised at the beginning. And then after I had looked at the same sort of problem with Beverly Balkow in looking at how migration would evolve, the rate of migration itself. So what I had discovered with the rate of recombination and the way it should be formulated as an initial increase problem, that led to basically a long series of papers that culminated, I think, there's still parts of it that are going on, but with the 2017 paper with Lee Altenberg in PNAS, it, I think, solves the problem in about as much generality as could be achieved. And I thought there might be a result like that when I suggested to Lee, when he was my PhD student, to try to get to that result. And we never quite got there in the paper in 84, in his thesis in 84, but he was persistent, and we recognized just exactly how important the paper by Carlin in one of those mathematics societies, long article on migration. And this article by Carlin had nothing to do with modifier theory. It didn't even mention the word, but it had some beautiful results on properties of some positive matrices. And Lee burrowed into that really carefully. And the result in 2017 was, I think, the definitive work on the reduction principle for migration, mutation, and recombination. A result that I was hoping for when I started trying to convince Carlin of my result in 1971 when I came back from Australia, which he didn't believe. And he had, I don't know if you know, at that time he did a lot of work with James McGregor, who had been a former student of his. And McGregor was hard of hearing. And so Sam Carlin would stand at the blackboard and do things like this and scream so that Jim could hear him. And our office, me and Bert Singer and Tom Leggett and Chris Chatrey, we were next door. We could hear everything that was going on. And so when it came my time to come back from Australia and show Carlin this result about modifiers of recombination, he said it can't be true. It's got to be wrong. He really screamed at me. And a few months later, he wrote the paper on the maximization of mean fitness principle, which said that the reason that the recombination would go down was because it increased the mean fitness. That was and he had 2 or 3 papers on that fitness maximization principle, which in the long run turned out to be wrong, because the mean fitness in normal population genetic sense is not an increase in function or decrease in function of the recombination.
Not with recombination, no. So,
but the argument that we had at that time, there was no way I was going to win that argument. New assistant professor at Stanford. Anyway, in the long run, he came around to agree with the result.
I think it was really a remarkable result, this reduction principle, just that many of these processes evolved to decrease, to make transmission more faithful, less recombination, less mutation, less migration.
Yeah.
That's a long time, though, between when you first realized that and when the big proof came. But when did you feel like you understood why it was happening?
When I asked in the beginning, I thought that there must be something in the mathematics of the local matrix that would force this to happen in connection with the eigenvalue to that matrix.
Mhm.
But I didn't know about that result that was part of variational theory. It turned out that how Carlin came across that matrix result, which was the result was obtained in a connection that had nothing to do with modification of anything. It just had to do with the properties of the migration matrix itself.
Mm-hmm.
Yeah.
It was Lee's astuteness that recognized that it actually could be transported into this formulation. And that I think is one of those things that people haven't recognized, just how beautiful that result really is. And yeah,
that body of work I think is well known, but maybe you were alluding to the fact that people could appreciate the insight even a little better. But I don't know if that's going to be your choice for this. We're wondering if there's a favorite paper of yours that you wish was better known. And is there an insight about that work that you'd like to share?
Well, the series of papers with Uri Lieberman on general formulations of migration, mutation, and recombination reduction, that's one stream. But I was so fortunate when I came back from Australia that Luca had been a relatively recent addition to the Luca Cavalli-Sforza I'm talking about, to the scheme of things at Stanford. And Sam Carlin and I, when I was a student and when I came back, we ran a seminar where we would ask people to come and talk about their ideas and their work, an informal seminar. And we asked Cavalli to come and give a talk about what he was thinking about. This was early 72. And he gave this talk about some ideas on cultural evolution. And he put up some statistics, which I actually I took the notes home and found that there were some errors in what he had put up. So I contacted him. He was such a generous guy, and he was so taken with the fact that I'd bothered to look into what he had said. So I wrote out what I had done, and so he said, that's worth a paper. So that was the first paper in cultural evolution that we wrote, came out in 73. And from that point on, for the next 35 years, he and I worked either 3 nights and a weekend or 4 nights and a weekend every week for 35 years. And that was how the theory of cultural evolution was born. And I mean, he had clearly had this idea from his time in Africa, where he had worked with the forest people, commonly called pygmies. And he was so engrossed in the ideas of how the different traditions would be maintained, communicated. And then we had the tremendous, I'd say, misfortune on the one hand and fortune on the other of having interactions with Jensen and Shockley as a result of the Jensen 69 notorious paper on how much can we boost IQ that relied on a statistic called heritability. And that led us to ask if the stuff that we've been talking about, namely cultural transmission, was important, then it would influence statistics about correlations between relatives. And that was how that paper in the American Journal of Human Genetics came to be published as a result of a lot of discussions and turmoil that was going on in the Bay Area around Jensen and Shockley. So I mean, that was the origin of it. But the paper that I think that I'm sorry has received not enough recognition was the one that originated gene culture coevolution. And that was the 76 paper that I did with Luca, looking at the coevolution of one trait that was involved in cultural transmission, vertical transmission, and a second trait that influenced that cultural transmission and in turn was being influenced by the cultural transmission. And the second trait was a diploid genotype. And when people talk about gene culture coevolution today, I think that paper originated that field, and it has not, it's not recognized as having done so relative to more conversational or narrative things about gene culture coevolution. And, you know, it's now, what is it, 50 years almost. And, you know, the papers like Sally's with Laurel Fogarty and other people's papers that that extend and look into how you would operationalize the parameters that we talked about in that paper to either be more realistic or give a better picture of what I call the gene culture disequilibrium and or pseudo linkage. Either way you want to call it, you just call it association, but it's the same kind of chi-squared relationship that I think has. I think that project has legs, and I think the paper that you did with that Sally did with Laurel is an example of that, and it's a nice area, and I think some of the techniques from a long time ago can be applied also to that.
Yeah, I have to say that I wasn't aware of that paper. We were digging into the early cultural evolution models, and then that 1973 one with Luca, popped out and was really relevant. And then, Mark, I will put on our YouTube page. We will put links to these papers so that they can
hopefully
really achieve the fame they deserve. And there's so much that math helps us understand about evolution, and yet I think so much confusion is in the field. Is there one topic or one area that you feel people are pretty muddled about, and you wish you could wave your magic wand and clarify?
There's a general area. Let me think about two areas. One that I was introduced to by Kevin Lala now and John Odling-Smee, which James referred to earlier, the niche construction stuff. It's now 30 years since they came down from Berkeley where they had been visiting, and we started to talk. And I think that many ecologists think that they already knew some of that stuff. I think that if they'd read Lewontin 1978 or the paper that he did, which 83, he has this paper in a journal where he talks about construction rather than classical evolution. And we cite that paper. He had those two differential equations. One, how the organism changes in response to both its history and the environment. And then he wrote another equation, how the environment responds to both the organism and the environment's history. Now pointing out that second equation didn't have in most treatments the organism in it. It was just the environment changed by itself, and organisms didn't change the environment. And it was eye-opening to me to see the possibilities that you could write a whole new set of evolutionary equations that impinged on ecological engineering and ecology in general. And John, I think, had that idea for a long time and was fortunate to hook up with Kevin. And I really have enjoyed my association with them since then. That was very important to me. Another relationship that I haven't mentioned yet, which was really important to me, was in 1973. Alan invited me to go with him to the University of Chicago, and we were going to stay there for a few weeks. And I got to meet, the first time I met Lewontin was right at the end of my thesis in 69. We had just done the papers. We just finished the work on symmetric viabilities, two genes with symmetric viabilities, and we had come across this general issue of how many equilibria could you get in a two-locus standard model. And we had looked at Lewontin and Kojima, which was inspirational. The paper of Lewontin Kojima 1960 was a tour de force. So I had the chutzpah when I went to Chicago to give a talk to say how incomplete Lewontin's treatment was. And I thought Lewontin might get really upset, but he didn't. He later on, he invited me to come to his lab, and I spent a month in Chicago with him, which was a tremendous eye-opener for me, the way he operated with his students with that big round table, the big oval table. You were there, you know that.
Yes, I know, I do.
And his flexibility in accommodating people from different disciplines. Mm-hmm. And then when he moved to Chicago, we actually got to write a couple of papers together. Well, the 75 paper in Science on heritability, and then later on properties of two-locus models. And it was eye-opening to me to see the way he interacted with his PhD students. And it was largely for that reason that I suggested that we swap students, and the people would go from my lab to his lab, like you did, Sally, and other people, and they would come from his lab to mine. And in fact, today, I'm going to be seeing Hamish Spencer from Otago, and Hamish was his student who spent months with me in my lab, and he's coming back for a few hours today. I'll get to see him again. But Lewontin's ability to absorb the wisdom of people from political science or philosophy was, to me, it was exemplary. And as you know, the people like Lisa Lloyd and Sober and Godfrey Smith, a lot of the leading people, if you went to Lewontin's lab, you would see them there. They were there learning about evolutionary theory and evolutionary genetics. And that kind of inspiration and flexibility, I would like to say, I would hope that I got a bit of that from Dick. And he was a bit hands off with students. I mean, if you look at the series of papers that they wrote, he's not on a lot of the papers that his students wrote, which is a style that he cultivated. He basically let the students go, like Marty Kreitman, Landy, those people who were very creative in their own way, and he supported them without being overriding, and that was a different way of operating. So I've got a great collaborator and way to deal with students.
Yeah.
I can think of that set of experiences and interactions with Lewontin. Maybe it could be part of this. I can think of other things you said that are relevant to this, but wondering, thinking about the perspective of a student entering the field, and this could be evolutionary biology or maybe mathematical biology more broadly. We're wondering if you have any advice about how to approach getting into this field and what insights theory and biology can provide.
Yeah. I find that there is a bit of a contradiction in the field, and that I see the field as very historical, and there is a way that the approaches and the topics build on one another from the 50s and 60s through to now. And I think that when I talk to my incoming students now, I give them papers from those periods to read to see how it was approached, what was left out, what's left to do, and what potential methods need to be applied. Not that the problem itself, but the way in which you approach the problem. And I think my advice and what I give my students now is some of these major papers from the past, to see the approach, the way it's approached, not necessarily the details, but the way that one approaches a essentially mathematical problem that has biological roots and may even have biological implications, and some of them can be important and some of them might not be. But I think that this reading of those papers and discussion of those papers, either with me or among the students themselves, can help a lot. And then if one encourages them to start writing early, then it forces them to start reading early. And that's one of the things that I think the communications revolution has resulted in, is that people don't read as much, and they certainly don't write enough in high school or undergraduate times to be able to say what's the introduction, the middle, and the end of a document, and the sequence of how it gets written, whether it's a purely theoretical problem or a problem that analyzes data. You want to make, when you write it with the reader in mind. And that's as opposed to writing it with you in mind and the writer. And that's one of the things that may be the hardest to communicate, especially because the way of writing, especially more technical stuff, is not taught in high schools anymore, and you're certainly not taught in most undergraduate. So I want my students to write early so that I can look at it and give advice from my point of view as an old fart in this business, you know.
I think also people make the mistake of thinking because it's technical, it shouldn't tell a story. Yeah. But if you don't have that narrative thread through your papers, then your reader won't get it.
Yeah, exactly. By the way, one of the influences that I had from Santa Fe Institute, probably the one that had the most, person that I met at Santa Fe that had the most influence on me, and actually interacted some with Sally, was John Holland. And John Holland was another one of those guys who was so general in his interests, and being in a psychology department, in a computer science department, and his understanding and appreciation of what the mathematics of genetics could tell you, and then how it could be reformulated in computerese, as it were. And he was really a powerful institutional figure at Santa Fe while during the time that I was closely associated with it.
And I remember when I got there to the Santa Fe Institute, your work, your cultural transmission, niche construction, things I talked a little bit about in the introduction, they were really in the air at that time. And so it's sort of clear your work has had an influence on the way people thought in that community. I guess John Holland seems like he was a big influence on on your thinking. I'm wondering, maybe even more generally, has your involvement with the Santa Fe Institute changed the way you think about how you do evolutionary biology?
Yeah, although I had appreciated the role of computational modeling, I hadn't fully appreciated it until I had these long interactions with John, and the genetic algorithms approaches showed me that there was another way that you could think about the same questions that we had been doing, and I know Sally was part of that too. And I'm very grateful to John because he put up with my incessant questions about where recombination was happening and how selection was working in the code that was being written for the genetic algorithms, and so I developed a much more tolerant view about the pure computational aspects that could be used for clarifying and initiating issues in evolutionary theory.
Because there, oftentimes it really is an optimization problem. It's a different problem than what evolution does.
Yeah.
It's optimizing.
That's another issue that that I wish I'd had a bit more influence on in the general evolutionary theory, is that the role of maximization or optimization has been a bit overblown, and we can approach almost every question, whether it's questions about altruism, cooperation, recombination, gene culture, coevolution, that the idea of optimizing something or maximizing something is a red herring, and you can it can prevent you from seeing the details of the structure of what's going on during the formulation of the model, and how the details of that structure prevents optimization from working or maximization. And if you decide to take a game theory approach, then the claim that it has to do with genetics or general biological evolution, I think, is in general wrong. And Hamish and I have written stuff on this too, but I think that people aren't understanding the limitations of that optimization or maximization approach to evolutionary questions.
I fully agree. And I think your work has really illustrated that what we have to think about is what can and does evolve, not what's best.
Yeah. And what are the parameters that would control that?
Exactly. Mark, you've had such an enormous influence on the field, but in particular through your students. You've had high school students, undergrads, PhD, postdocs that have gone on to shape the field. I think you must have had more students that you've really guided than any other person in evolutionary theory, and maybe in evolution as a whole. How many students have you had?
I once again, I think there are two things that I would say I've been really lucky with. One is the very first two students I had, that one came with me from Australia, was Glenys Thompson, who became a professor at Berkeley eventually after a postdoc at Oxford with Bodmer. They're a really brilliant student. And having brought her with me to Stanford was, I think, one of the best things that I did in that move. The other thing was the first two postdocs that I had at Stanford, I was introduced to one of them by Timothy Prout, who was then at Riverside, but had spent time in Denmark with over Friedenberg at Aarhus, and he arranged that I meet Freddie Christiansen. And Freddie came to Stanford as a postdoc. And basically for the next 10 or 15 years, we collaborated, and he collaborated with you too, Sally. And Freddie was just a marvelous guy, a sad loss too early for his departure. And Freddie was very flexible. He lived at our house for 6 weeks every summer, and his insights into statistics in particular and how they apply to evolutionary questions were really formative. And then Yossi Hillel, who was an agricultural geneticist from Israel, also spent 3 years at my group in that very early period in the early 70s, and introduced me to a whole other set of issues in plant evolution and statistics. And the good fortune that I had to have those people come so early and sort of introduce the breadth of what you could be done in the field.
Mm-hmm. Mm-hmm.
That is, I think, salutary, really important. And then, it became really nice that Stanford was starting to get some recognition as a place where people should go to do their PhDs. And then people like Andy Clark and Ron Burton and Kent Holsinger and people who were very independent in their own research, but had envisaged extra value in coming to Stanford. And not only did they benefit from it, but they taught me a huge amount. That sequence of students was really critical for me, and it became part of my life that the interaction with students would be so stimulating and rewarding that it became what I think is the most important thing that I've done.
It sounds like you have had not just a large number of students, but really deep interactions with those students and learned a lot from those interactions. For somebody who might be just becoming an advisor of students at this point, can you distill that into any tips or guidance that you would give?
Yeah, I think that appreciation of the fact that the students are going to give you a lot, that it's not a one-way thing, that even from the beginning, knowing that every one of the students is different from every other one, and then trying to find what their really strong points are and cultivating those strong points to the maximum and having them appreciate what their strong points are so that they can make the maximum use of those strong points. And getting them to put their thoughts on paper early so they get to know how to write the papers. It doesn't have to be anything publishable, but just trying to codify your thoughts and put it in a more or less rigorous scientific framework that most people don't get as undergraduates, the experience to do that.
Yeah.
Some do, and those ones are very fortunate, but then to have them interact with one another, it seems to me that the interactions between graduate students are really most productive. And I always remember Sally's interaction with David Goldstein as one of those, but there are always interactions that go on. And I think if you have the students close together physically in some kind of situation like we used to have in the now departed Heron labs at Stanford, but even now where we're in a more industrial kind of construct for the lab, the students are interacting with one another pretty much daily. And you've got to kind of get an optimal amount of interaction so they actually don't spend the whole time talking about what happened over the weekend. But they're actually discussing what they read or what they're about to write. And I've been unbelievably fortunate in having that cadre of students who wanted to come to Stanford, and having other colleagues in the Department of Biology that were sympathetic for a long time to me being the only kind of person there who did sums, as it were. And then having being able to bring in Noah and Jonathan Pritchard, truly great scientists. And Tolja Prakar. So the critical mass of theoreticians has been achieved relative to what I could call the norm in biology departments. And so the students can get a lot from any of those people. And I encourage them to do that, to interact with the students of the other professors, and go to their group meetings, their seminars, and learn what's on their minds with respect to what other priorities to do research on. And if they choose that, then that's fine with me, as long as it can advance their learning and what they're going to do after they leave.
That's really great advice. And, I think your mentees would remember those times where you were so supportive and really you listened. Yeah, even when there were crazy ideas coming out of our mouths, you helped us figure out what the sense of those ideas were and how we could make those rigorous.
I'm not a believer that many ideas are crazy. I think there's often some germ of substance, no matter how crazy it sounds. Listen and get a bit of that to see and not be narrowly defined. Mm-hmm.
Right. Marc, I think the fields have changed quite a bit in the relationship between modeling and biology and the perception of theoreticians, the people that do sums, since the start of your career. Any reflections on that?
Yeah. There have been people who did sums, but not rigorously for a long time. And I think it became a question of more, I would say, an intellectual divide between those who did sums not rigorously and those who did them rigorously, rather than who did sums at all. And that I think is somewhat dominant. I think it may be somewhat cultural. It may have to do with a school on the one hand that I would put Carlin and Ewan's in, and the English school on the other hand that followed Hamilton and maybe Maynard Smith, that wasn't so rigorous in the kind of sums that they did, the kind of mathematics that they did. And both groups were doing theory. But I think that there was a conceptual divide, certainly an appreciation divide between the two kinds of theory, and I think it still exists. I think it's most probably manifest now in the fields of cultural evolution. But and I think the value in pure population genetics of the rigorous mathematical theory has been proven. And so I think it's less, the cultural evolution stuff needs another few years for that to solidify. But I think when we started in the beginning, I remember thinking with Carlin, we would sit down and think about theoretical population biology what would the first issue consist of? The first issue of 1970. And it was rather difficult before that time to get the pure mathematical stuff published. There were very few journals that were that sympathetic to it. And that was the reason that we decided to approach academic press at the time. It evolved into other things, but and they fortunately saw the value of that kind of thing, even though there had been a few major scientists who had published a relatively pure theory. But it was really important to get the ecologists, demographers, and epidemiologists on board, the ones who are doing really innovative stuff like MacArthur and Nathan Kieferts and those people who were leaders in their theory, in the theory that was applied to their kinds of issues. And fortunately, they saw that there was a value to this. Today, it's not clear to me that the value of the pure theory is being appreciated quite as much. I'm a bit worried about that. And a part of it has to do with how little statistics and math a lot of the people who are not doing theory actually know. And that makes it difficult for them to appreciate what goes behind making statistics and making expectations for things that you can quantify. So I think it's been a problem, and some of it, of course, has to do with different mathematical approaches. I mean, there's a way physicists approach the mathematics and the way mathematicians or statisticians approach the quantitative stuff. And that can also be a roadblock to mutual understanding. I'm mildly optimistic that dichotomy will be resolved over the next few years. And this Society for Mathematical Theory in Population Biology might be able to contribute to the resolution of that somewhat of a dichotomy.
Yes, my students and I are reading a book about modeling and approximations from physics, just something we hadn't really learned. So I agree with you. We need to bridge. You know, computers to some extent, I feel like they've allowed so many biologists to bridge these subjects, but maybe something you said makes me wonder if, yes, they enable more crosstalk and more people can get involved in theory, but perhaps the appreciation of what analytical proof gives is declining.
That's right. That's a good point. I remember that when I joined Stanford, when you join a university, they ask you to specify what kind of a lab you want. So I asked for a computer, and so they allocated an office for the computer. IBM 1620 occupied a whole office with the punch cards and all of that stuff. Sadly, a couple of years after I got that, there was a fire in that office because the air conditioner, which you had to have in there, separate air conditioner, caught fire and destroyed the computer, which may probably have been a good, what I would call a forecast of what computers should be in the future. but anyway, that was a rather salutary occurrence. But we did use it quite a bit. And people started, I would guess, by the mid-70s, were using more and more computational tools. And they can be really informative. And they've gotten to the amount of time that you have to take to do the parametric explorations to find the generality of what you're trying to say. save a huge amount of time on that and can actually lead you to findings. So one set of parameters that you couldn't really investigate by hand might have a different result from some other set of parameters. And that could lead to a question, why is that the case? What is it about the structure of the underlying model that let this happen? So you might not have been able to discover that. It's almost like going out in the field and finding an anomalous animal or something that shouldn't be there, that kind of thing.
But then there's a role going back the other way, isn't there, to kind of understand, well, what can the model tell you now? You've sort of seen there's something there. What is it that's there?
Yeah, that's correct. What does it mean if you have a discontinuity of this kind, or you have an extra fixed point of that kind, or you have an eigenvalue that is a function of all these parameters? How do the parameters interact? And that's very true. Mm-hmm.
So I have a question about now. I think you mentioned the role of random chance in your trajectory, not just in the topics you're working on, but in the trajectory itself. So I don't know if random chance is going to be the answer here, but you have worked on a very broad range of topics. Human genomics. We've talked about cultural evolution and niche construction, a really broad range of topics. And so, how do you identify what are the good problems to work on? What the next thing you're going to do is?
One way to do that is to have invitations to people whose work you don't know very well to come and give talks, and they open your eyes to questions which you might not have thought about before, and then talking to them enhances the novelty in your own mind, as well as the possibility of going a bit deeper with that person, finding out what they really had in mind and what they expect from their stuff. There's nothing better than reading also. And I read a document that a colleague here at Stanford named Jessica Riskin, she's a historian, and I got a copy of the book she wrote called The Restless Clock, which is a book about the early foundations of mechanistic thinking in biology and how it might have had influence on some of the later evolutionary people in this 18th and 19th centuries. Yeah. And so I had asked her to come over to do a talk in my group, and I hadn't met her before. And we ended up writing that piece in the New York Review of Books, which reviewed Hardin's book on the genetic lottery. And we've now been collaborating ever since. And she has a huge amount to teach me. And she's got a wonderful new book that's coming out later this year, early next year, on Lamarck. And I learned a tremendous amount from that. And it's clear that I didn't know that Lamarck's work on giraffes was quite late in his career. Yet it's the thing that everybody remembers, right?
Right.
But it's not the main thing that he did. There were lots of important stuff he did before that. And how it relates to today's evolutionary theory is really quite important, in my view, that there's a lot that we can learn. And I think some of what we did in the book with Kevin Lala, the new evolutionary book, Evolution Evolving, there's some positions in that book that we take on the role of development that you could say are quite Lamarckian. And I know that one of the criticisms that anthropologists made of the work that Luke and I did on cultural evolution was, you're really Lamarckian. The individuals are transmitting their environments. Okay, so that didn't faze us one bit, I must say, that we didn't regard it as a negative.
Right. Right. If anything's Lamarckian cultural evolution ought to be. So Mark, your career started more in kind of the statistical math biology, but we're really seeing an explosion in how much statistics, bioinformatics, AI, machine learning, all of that is happening. Do you think that theoreticians of the future will use different tools, um, or these approaches that have been tried and true in the past will continue to be important?
Yeah, I think they will. Even the high school people who are in my group now they're quite familiar with some of these computational tools, and they use them to make even small inroads into the questions that they're asking. And I mean, there's no doubt that AI has had a big influence on molecular approaches now. There are so much data and so much to learn. I'm a bit worried that people might lose the ability to analyze logically themselves if they think that just by training this program to recognize these features and then having it recapitulate those features in some new data set, whether it finds them there or not. It's a form of thinking that may not be rigorous enough. And I think there is a role for rigorous thinking, for rigorous logic to be incorporated into our field of theoretical biology, whether it's ecology or epidemiology. That there's a role for that kind of rigor. And I think probably Warren Ewens would agree with me on this. I would hope he would. And the kinds of questions that he asks about under what conditions does Fisher's theorem really apply, or what is the meaning of effective population size? Those kinds of questions. You can't get the answers to those by artificial intelligence or some computational analysis of data. They require some quite rigorous thinking.
Mark, that's a really great place to end this conversation. I think you've been a leader in training students and establishing that rigorous thinking. You've led the journals that establish that way of rigorous thinking in biology, and we really appreciate having you have this conversation with you today.
Okay, well, it was kind of nice to think about it, and I hope I haven't offended anyone.
Hopefully not.
Okay, nice to talk to you. We'll be in touch.
Thanks, Mark.
Bye-bye.